Okay, now we're going to discuss section 6, chapter 2, talking about water mixtures, comparing and contrasting the three different types of water mixtures, explaining how an emulsion differs from other types of mixtures, and explaining the different ways to express the concentration of a solute in a solution. Starting first with the idea of a mixture, which is formed when simply combining two or more substances. Two defining features: substances are mixed but they're not chemically changed, and they can be separated by physical means. An example would be evaporation or filtering. Categories of water mixtures, there are three. Suspension, which is a material larger in size than a 1 millimeter mixed with water. Example would be blood cells within plasma or sand in water. It doesn't remain mixed unless it's in motion, and appears cloudy or opaque, and it scatters light. Okay. A colloid is a smaller particle than a suspension, but it's larger than those in a solution. An example would be fluid in cell cytosol, and the cytosol of the cell, and fluid in blood plasma. It remains mixed but not in motion, and it scatters light. Okay. And then a solution is a homogeneous mixture of materials smaller than 1 nanometer. It dissolves in water. It doesn't scatter light, it does not settle if solution is not in motion, and it's more an example of sugar water, I should say, not sugar and water, but sugar water, salt water, blood plasma. And a special category of suspension is emulsion, whereby water and a nonpolar liquid substance are combined or together in that category of suspension, and its an example would be oil and vinegar salad, dressing, or breast milk. It doesn't mix unless it's shaken. Okay. Now, so we have an example here visually of suspension, blood in motion. You can see it here, and when not in motion, you can see the plasma, the leukocytes and erythrocytes. They scatter light and they settle if mixture is not in motion. That is suspension. Colloid, smaller solutes scatter light but do not settle. And solution, smallest solutes do not scatter light or settle. And then emulsion, the special category of solution, like oil and water, right? In motion versus not in motion, in which case the polar substance, the water, and a nonpolar substance, the oil, form an emulsion when agitated in motion. Okay. Concentration is determined by the amount of solute dissolved in a solution, which makes sense logically, right? The expressions of concentration, essentially there are 4 expressions of concentration. Mass and volume, mass over volume, the mass of the solute per volume of the solution. That's like the results of a blood test. Mass over the volume of percent, like the grams of solute per 100 milliliters of solution, like IV solutions. Expressions of concentration, the final 2 more. Molarity, which is the moles solute per liter of solution. It alters with the changes in temperature. And it's more easily measured in the body than molality is, and molality makes, or I should say, is described as moles solute per kilogram solvent. It doesn't alter with changes in temperature, and it's slightly more accurate than molarity. Osmoles is a unit of measurement for the number of particles in a solution. It reflects whether the substance dissolves and dissociates. It can be expressed as osmolarity or osmolality. Osmolarity is the number of particles in a 1 liter solution. It's easier to measure. It's like blood serum expressed as milliosmoles. Osmolality, the number of particles in 1 kilogram of water. It's more accurate, but it's difficult to measure. Okay. 1 mole is essentially 6.022 times 10 to the 23rd atoms, ions, or molecules. Big number, right? The mass in grams is equal to the atomic mass of an element or the molecular mass of a compound. 1 mole carbon is essentially 12.01 grams. Just so you know, you're not going to be tested on these exact numbers for amounts. You have to remember these amounts. The ideas, yes, not the amounts. Okay. To find a molecular mass, multiply the number of units of each element by the average atomic mass and add the totals. And there's some variation due to isotopes, right? Isotopes having variation between an element with a variation in terms of being an isotope. So, when left standing, erythrocytes, or red blood cells, settle to the bottom of a tube of blood. Based on this characteristic alone, blood would be characterized as A, suspension, B, colloid, or C, solution. The answer is A, suspension. Why is blood also considered the other 2 types of water mixtures? Well, blood is mostly water, right? Blood plasma in the blood, which is the largest substance in the blood, is about 55% of the blood, and 90% of that is water. So you got basically half water in blood, right? What are 4 ways solution concentration may be expressed? We have mass to volume, we have molarity, we have molality, and mass to volume percent. Okay, and that is section 6.